Okay, good morning, everyone. I'm Rob McNamara. I'm from the University of Cincinnati, the Department of Psychiatry, and I have come to an appreciation for omega-3 fatty acids from a slightly different perspective. And what I want to present today is sort of a translational story in a nutshell, because 15 minutes, I could spend 15 minutes on each individual slide, actually. But I'm an academic, so forgive my monotonous, less flamboyant presentation than Joe. But so in academia, we need to disclose people that fund you, and so I think this is testimony to a growing interest, I think, at all levels of the potential role of diet and fatty acids in psychiatric illness. Otherwise, I mean, this is really proof you're not going to get funded. And so some of the tenements of why I'm very interested in and became interested in the first place in omega-3 fatty acids is that, importantly, DHA, one of the long-chain omega-3 fatty acids, is very abundant in mammalian brain. In our postmortem work, it's around 14% of total fatty acid composition. Unlike EPA, which is very low and often undetectable, but that's not to say that it doesn't impact the brain from a peripheral mechanism, which I can discuss in a bit. As we're beginning to understand, preformed DHA is arguably the best source of DHA, given that the biosynthesis from ALA is so limited, and indeed from EPA is very limited. So we know that DHA, two things are important here. It's the most abundant omega-3 in the brain, and fish and seafood is really one of the few sources that are available other than supplements. The other thing that's of interest to us within the context of psychiatric illness, this is really difficult to see, but we know that based on retrospective studies that the initial onset of, I'm going to talk a lot about bipolar disorder today because that's really epitomizes mood swings. Initially, the initial age at onset is during adolescence and in children. Most people don't recognize that. 76% experience their first onset of a manic symptom, and by definition bipolar 1 disorder, below the age of 21 years of age. And so it's of interest, therefore, that during that period, this laser is useless, there's a dramatic and rapid increase in DHA composition in the postmortem human brain. And this is a Carver study, which a lot of people cite. This is a difficult study. These are healthy, albeit dead, subjects. But it's these are actually very difficult studies to do. And but what's important is that we see the exact same thing in rats, in the prefrontal cortex over the equivalent period of adolescence. And importantly, in rats, we can selectively manipulate the DHA accrual that occurs during the adolescent period and see what that does on a number of different endpoints. And I'm going to be talking about some neuroimaging endpoints because the approach we're taking is to be as translational as possible. Really, the ultimate objective I have is to convince entrenched psychiatrists who have sort of adopted and are unyielding about it, sort of a pharmacological approach. And as I'll discuss, that is inadequate and in fact might be harmful to people that are at risk for bipolar disorder. I'd also like to mention that It's not the case that DHA just gets into the brain and sits there and becomes part of the structure of membranes. While that is true, we also know that there's dynamics involved, and DHA can be released from membranes and therefore liberated to become a substrate for anti-inflammatory molecules. And this is particularly important within the context of bipolar disorder and depression, which we believe, or we're coming to learn, is associated with sort of unresolved chronic low-grade inflammation, which we think is perhaps one of the mechanisms by which omega-3 deficiency may contribute to neuropsychiatric illness. So I'm going to go through, bear with me, some rat studies, and then I'll get to, I'll translate, well, actually this isn't rat. So the other important point is that the adolescent period, we know with some certainty now, due to a lot of work done at NIH, is a period of rapid pruning that occurs in the prefrontal cortex. Forget it. And so if you look at the top, so over the course of adolescence, there's a significant reduction in gray matter volume, which we, through postmortem studies, appears to be due to synaptic pruning, while at the same time there's a significant increase in white matter maturation. So this is, adolescence is a very dynamic period for the prefrontal cortex, which we know is very important for attention, executive function, as well as mood regulation. And there's a growing consensus that bipolar disorder is associated with deficits in prefrontal cortex activation and concomitant elevations in amygdala activity in response to emotional responses. So people with bipolar disorder tend to overreact, or there's a lack of inhibition over their amygdala response to emotional processes. And similarly in rats, we know with greater certainty that the adolescent period is associated with approximately a 50% reduction in the connections between the prefrontal cortex and the amygdala. So this is well defined, and so we sort of capitalize on this information and attempt to understand what are the effects of varying omega-3 diets on a number of different endpoints, as I mentioned. And so one of our models is simply, well, essentially 3 diets. One is a control diet, which has ALA in it, and this is standard lab chow, really. Our second diet is deficient, in which there's no ALA in it, and this leads to reductions, which we believe are more clinically relevant than the perinatal models. We're also doing perinatal work as well, which I don't have time to discuss. And then we have a fish oil diet, which essentially replaces the ALA with fish oil. And when this creates a spectrum, this is the prefrontal cortex fatty acid composition in rats. And I put this diet composition up there because it's important. One of the things that you are afforded when doing animal research is you can tightly control other dietary components which represent potential confounders in human research. And so all of these diets have the exact same vitamins and minerals, et cetera. So the only thing that's changing is the single fatty acid, essentially. Okay. So one of the things that we found is that deficient animals with these moderate reductions in prefrontal cortex DHA levels exhibit significant reductions in what's called radial diffusivity, which is a term to indicate that in the white matter, it's an index of white matter microstructural integrity, which is gaining favor, certainly within psychiatric research. And it's indicative of, vis-a-vis other models, a dysgenesis of white matter. We're not sure why it's lateralized. We were actually just happy to see something, but this indicates that omega-3 deficiency may contribute to perhaps a delay in the maturation of white matter in the rat forebrain. Other studies have looked at the neurochemical effects of omega-3 deficiency, and something that we recently published was the effects of the diet on glutamate concentrations in the prefrontal cortex. And as we were writing this up, there was a meta-analysis that came out in JAMA Psychiatry arguing that, or demonstrating that, at least in schizophrenia, that, and there's also data in bipolar disorder as well, the glutamate elevations in the prefrontal cortex appears to be part of the pathophysiology of schizophrenia. So we can actually recapitulate that in a rat by simply making it omega-3 deficient over the course of adolescent development. And there's other subtleties. For example, it appears to be mediated in part by reductions in the astrocyte glutamate uptake transporter. And we do know that omega-3s do play an important role in astrocytes as well, and that's which play an important role in neurovascular coupling, which I'll get to in a minute. This is complex, essentially the same design, but in this case we looked at the effects of chronic amphetamine exposure, which we use in kids to improve their ADHD, and we believe that its efficacy is due in part to a activation of the prefrontal cortex, which then acts as a brake on the rest of the brain. And what we found is that chronic amphetamine did indeed result in an increase in the response to amphetamine in the frontal cortex of rats, and that if you're omega-3 deficient, you just didn't see that recruitment process. And we know from other studies that the chronic amphetamine is associated with neuroplastic changes within the prefrontal cortex that are quite striking, and omega-3 deficiency appears to impair that. And I think the clinical upshot of that is there's some evidence that omega-3 adjunctive supplementation may actually augment the therapeutic benefits of stimulants in kids with ADHD, and this may represent one mechanism. Okay, enough about rats. Now we're moving to our closer cousins. So this isn't my work, but it's when I saw it, I was very excited. It was published in Journal of Neuroscience, which is a top-tier journal in neuroscience, and this is from the Neuringer group. And what they found, which was of significant interest to me, was that using I don't have time to get into the details, but they saw reductions in functional connectivity. This was resting state connectivity between the prefrontal cortex and other prefrontal areas. And this was of interest to me because, being in psychiatry, there's been a number of papers suggesting that bipolar disorder is also associated with a sort of a disconnectivity. And as I suggested earlier, connectivity between the prefrontal cortex and the amygdala may be a very important part of the circuit. Okay, now to humans. And I'm going to describe a couple of cross-sectional studies that stemmed from an intervention study that was originally sponsored by Martech. But we had all this incredible data set that we were able to go back in and do some data mining. I'll have to be very quick here, but we, in essence, found that, so we had approximately 50 kids, and we divided them into low versus high red blood cell DHA levels by a median split, and just compared those two groups. And one of the things that we found was that we saw reduced functional connectivity during an attention task with some of the same regions that I just alluded to in the monkeys. So we can recapitulate that also. And these are kids that are 9 to 10 years old, healthy, although we did find that those that were in the lower DHA group had slower reaction time on the sustained attention task, which has to do with processing speed. And so this is an MRS study looking at neurochemicals, and this was quite a striking finding to us, that if you just look at low versus high DHA levels, we see significant differences in a number of neurochemicals. And what was striking to me is that this in some ways was a mild version of what we see after sort of an ischemic insult, which led to us hypothesizing that perhaps a mild reduction in blood flow to these regions may be mimicking the sort of the neurochemical fingerprint. Okay, 8 seconds. So this is the intervention study that's been published for some time now, but it is interesting because it, if nothing else, demonstrates that you can intervene in these kids and significantly increase their blood levels with DHA. And one of the main findings, given that I have 0 time left, is that during an attention task, the kids that were given the highest dose of DHA exhibited significant increases relative to baseline and relative to the change in placebo from baseline. Significant increases in bold activity, which is ultimately reflective of neuronal activity and blood flow combined, suggesting that there's an improvement in neurovascular coupling during performance of an attention task, which we specifically chose because we know it activates the prefrontal cortex. We're starting to move this work into individuals with psychiatric illness, kids with bipolar disorder. This just happens to be a incidental finding that we haven't published, but we saw significant correlations, positive correlations between red blood cell DHA levels and prefrontal cortex activation in response to attention in kids that were depressed. Another finding, this is just red blood cells now, we have demonstrated, we have observed that kids are at high risk based on having a biological parent with bipolar disorder and having an ultra-high risk, UHR, having a biological parent with bipolar disorder, and also having active depressive symptoms. And this is a first episode bipolar group, all adolescents. And one of the things that we found was that the increasing risk as we move towards full onset of manic symptoms and bipolar disorder, we see a decline in omega-3 levels, suggesting that this may be, if nothing else, a risk biomarker. And importantly, we found that there's correlations between the red blood cell DHA levels and EPA levels and mood symptom severity, both manic symptoms as well as depressive symptoms. And so perhaps one of the upshots of this, and as I alluded to in the beginning, is what is the role of omega-3s in clinical practice? And very briefly, although Bill's not here, right? Is Bill going to give a talk?
He's going to give his talk.
Oh, I'm sorry. Sorry, Bill, wherever you are. So we know that SSRI medications in this high-risk group can actually precipitate the onset of manic symptoms, and therefore you have a diagnosis of bipolar disorder. And so the question is, are there alternatives that are less problematic for this high-risk group? And fish oil comes to mind. And we completed this study, an NIH-funded study, where we actually observed significant reductions in global measures of improvement by the physician. We didn't see significant reductions relative to placebo on our primary measure, which is the CDRS, Depression Symptom Severity Scale, but that's actually not too surprising given the power of placebo. But we also found that at endpoint, we saw significant baseline to endpoint reductions in limbic structure activation in response to an emotional stimulus in the kids with the placebo, but in the kids with the fish oil treatment, but not the placebo. Obviously don't have time to get into this now, but reductions in cerebral blood flow may represent a mediating mechanism by which omega-3s may help contribute to not only healthy brain development, but also potentially improvements in symptoms and brain activation and brain intercommunication. And Tom alluded to this because you remember it from back in 2004. But this is, I think, would be a good advertisement on a brochure of some sort. All right.